The area of molecular oncology is advancing at a very rapid rate. We now have an armory of molecularly targeted therapies and immunotherapies that we can prescribe to our patients. In fact, with the most recent approval, there's 15 in lung cancer alone, compared to just 1 10 years ago. The challenge for molecular pathology is now that we need to analyze a broad variety of biomarkers, and we need to do this analysis on smaller and smaller amounts of tissue. This tissue is often highly contaminated with normal cells, so we need to do this in a highly sensitive manner, so that when in these small biopsies, when they only have small amounts of cancer tissue, we can still assess for these important biomarkers. And then, of course, we're now in the era of treatment monitoring and using liquid biopsy to look at the resistance mechanisms in cancer in real time so that we can be changing therapies, applying alternative therapies at the right time point to give benefit to our patients. My name is Darryl Irwin. I'm the Senior Director of Scientific Affairs at Agena Bioscience. And it is my pleasure to introduce to you our two speakers today. Our two speakers are going to talk on those two different topics. Firstly, tumor assessment, so assessment of solid tissue for those biomarkers, and then second, in liquid biopsy. So our first speaker is Dr. Eduardo Castro-Echeverry. Dr. Castro is a board-certified anatomical pathologist and clinical pathologist with certified subspecialties in the areas of hematopathology and molecular genetic pathology. Dr. Castro is currently a staff pathologist at Baylor, Scott & White in Temple, Texas. Dr. Castro has recently done a validation study of Agena's IPLEX HS chemistry and is going to talk to us about the rationale for selecting that chemistry and the results that were achieved in that study. So thank you, Dr. Castro.
Excuse me, let me just set a timer, make sure I don't go over my allotted time. Thank you, Darryl, for the kind introduction. My name is Eduardo Castro-Echeverry. I am a staff pathologist at Baylor Scott & White Medical Center. And it's my pleasure to be here to talk to you about our experience with the Agena cancer panels. So first, I should do some disclosures and talk to you about some of my conflicts of interest. This is from the, based on the Sunshine Act. This is what you can pull up on me if you go to the website right now. I am actually right here, and most physicians you will see are right here. So, so where does that $127 come from? Whose pocket am I in? Well, so I am in, apparently Roche bought me a meal at some point in time, and I don't remember that meal, to be quite frank with you, so I don't know what to make of that. So the some other disclosures and conflicts of interest. Agena did offer to pay for the hotel and travel expenses to give this talk. I declined. During the purchase of this equipment and subsequent validation of this panel, I actively lobbied against it, and I'll tell you why at the end. And when I was invited to give this talk, I accepted because I was invited to attend it. So you should all read your emails. All right, let's keep going. So let's talk about who exactly I am. So Baylor Scott & White is one of the largest healthcare providers in Texas. When you hear Baylor, you might associate it actually with the, sorry, where is it? All right, with the Baylor College of Medicine, which is down to your bottom right corner. A lot of our nation's finest physicians came out of the Baylor College of Medicine in Houston, Texas, but I am not them, so that is not me. Baylor Scott & White Medical Center is the product of a merger between the Northern Texas Dallas Division and the Central Texas Division. We have not merged our pathology departments, so I am not the North Texas, I am the Central Texas Division. Okay, so now we know who I am and what I do. So this is the NCCN cancer treatment guidelines for non-small cell lung cancer. This is what our clinicians face when they have to evaluate a patient who's been newly diagnosed with non-squamous cell lung cancer. And the main reason I put this up here is to show you that genomic profiling of lung cancers is now part of our treatment guidelines. Right? Here, BRAF, where else? I believe there was some KRAS there. But, and then as well, there's some fish-based assays that we need to do, ALK, ROS, as well as some PD-L1 studies. But our clinicians do expect us, as part of the standard treatment for patients who are newly diagnosed with lung cancer, to be able to evaluate the genomic profile of any given lung tumor. And the treatment guidelines also include some emerging biomarkers that also have treatment implications. Same for colon cancer. Here we see, this is where we see our RAS and BRAF and KRAS make their appearance. And again, our clinicians, in order to treat their patients based on NCCN treatment guidelines, expect us to provide them with genomic information. So what is the molecular pathologist to do? What can we do? So we can do NGS-based assays, and I am not an expert on mass spec. My background is in NGS. I am most comfortable looking at a pileup on an IGV screen. That is what I like. So I like NGS-based assays. You can analyze for a lot of different genes at the same time. You can do individual gene sequencing, so you can do Sanger or Pyro sequencing, but once the gene list that you're after starts getting bigger and bigger, it starts to get more expensive and more onerous. You can try some other strategies. Those include the Agena MassArray using mass spec. You can try allele-specific PCR, or as a regional molecular pathologist, you can send it out to another institution. This is not a popular choice typically because it means that you are basically sending that business along to a different laboratory. So our NGS-based assays, they come in all shapes and sizes. There's any number of genes. You can do anywhere from 15 to 500 gene to whole exome, whole genome, fusions, copy number variants. The analysis can be scaled and automated. You can reliably detect variants at VAFs of 5%. If you want to go lower than that, things get a little bit more dicey. But there's some assays out there that will tell you that they can detect variant allelic frequencies lower than 5%. And you can evaluate cool things like subclonal populations and clonal evolution of your tumor. On the other hand, it's fairly expensive in terms of the machines, the samples required for validation, the training required to get it working. Validation can take a long time. Per CAP guidelines, you need 59 samples per variant type, and while you collect all of these tumors for the validation, well, that takes time. You need some bioinformatics support. This, for us, and I'll tell you that right now we're working on validating some NGS-based assays for our tumors. This has been the biggest challenge. So about a year ago today, we decided to go with a bioinformatics company to evaluate our FASTQ files. And between us deciding that we were going with that bioinformatic company to our institution approving the bioinformatic company was 9 months. Now, in those 9 months, you are expected to provide genomic information for all of your patients' tumor samples. And what are you supposed to do in that meantime? Turnaround time can be an issue. Single gene sequencing, it's excellent if you want orthogonal confirmation. If it's worth considering if you're looking at very limited gene panels and low volume, but it does become unwieldy as more samples are involved, and it's very, very challenging at low variant allelic frequencies. So the Agena MassArray platform is a MALDI-TOF based assay. It's the way it works. There's ionization of your DNA fragments. Then alleles are separated based on time of flight. Your basic workflow is you do PCR amplification of your enrichment of your sample. There's a primer extension, and then the sample goes through the mass spec, at which point you obtain your data. The workflow, I will tell you, is quite easy. So our techs are quite happy with the ease with which they are able to set up this assay. And the hands-on time is about 28 minutes, and the time between starting the assay and getting some data is about 8 hours. For the sake of comparison, the NGS assay we were working on that we ended up not going live with was the TST15 from Illumina. From their website, the amount of hands-on time is about 3.5 hours, and the time between DNA to data is approximately 36 hours. And we're talking about a limited 15-gene panel, okay? So the Agena Lung Panel covers all the genes involved in the NCCN treatment guidelines. It includes BRAF, EGFR, ERBB2, KRAS, and PIK3CA. It's actually selected exons and codons within those genes. Now you may ask yourself, is this enough for your lung cancers? And that's a fair question. So what I did was I took a look at the data and saw what exactly we were missing, because that's an important question you need to ask yourself. So this is from the CBio portal website. This is managed by Memorial Sloan Kettering Cancer Center. This is publicly available data on all the cancers that have been sequenced by Memorial Sloan Kettering. And this is the breakdown, or the percent breakdown for, the percent breakdown for our ERBB2, KRAS, EGFR. So this is the MassArray panel on the left, and the percentage numbers is the percent of non-squamous cell lung cancers that are covered by that gene, or that have a variant that involves that gene. So certainly we're not at 100% with the Agena panel. Actually, we're closer to 60% or so, and if we're being rigorous, the truth is the Agena panel does not cover amplification, which is your red bar, covers mutations, which are your green bars. So we're about 40 to 50% coverage, but certainly all the genes that are relevant for NCCN treatment guidelines. Also worth noting that it does not cover all the relevant codons for your KRAS gene. So this is, again, from our helpful OncoKB website. So our Agena MassArray covers here, and these are, to be fair, the most common variants that we identify in these tumors, and the ones for which we have the most clinical evidence. So certainly you're covering the lion's share of what's out there for KRAS, but there's some coverage that's not included. And the same for EGFR. Again, we are covering not everything under the sun, but we are covering the lion's share of the mutations involved in EGFR. That's the Agena colon panel, similar story. It has it's a 5-gene panel with mostly the relevant exons included. And if we look at the data from Memorial Sloan Kettering, I apologize, those numbers are way too small, but here they are bigger. So again, we're not at 100%, we're more at approximately 70% for the genes of interest. Now, sorry, that is duplicated. Okay. So when we went to validate, let me tell you a little bit about the validation of the study. So we evaluated 20 samples, including 18 lung cancer samples from formalin-fixed paraffin-embedded tissue. We manually microdissected for tumor enrichment, and also included 2 non-template controls. All tumor samples had been previously characterized by pyrosequencing, which was our method of analysis prior to adopting the Agena. The peak intensity, we'll get into why this is important. The minimum was 5, but the z-score we picked initially was 35. As far as the lung panel validation, we had for KRAS, we had a positive agreement of 98% with using the pyrosequencing as a gold standard, and a negative agreement of 71.4%. So what does this actually mean? Means that there were Agena called variants that were not initially called by pyrosequencing. And so we had to investigate those further. For EGFR, we had a similar situation, positive agreement of 95%, but there were 2 variants that were identified by Agena that were not identified using pyrosequencing. And a similar situation with the BRAF gene. Now, taking a closer look at those discordant results. So here's our list of validation samples. And if we look at some of the discordances, so when we talk about findings that were not seen in the pyrosequencing but were identified in the MassArray, so that includes this EGFR insertion. It also includes this PIK3CA mutation. And it includes, there's also some additional discordances. So there's also this KRAS. Sorry, I'm not very good with the pointer. There we go.
Yes, yeah.
There we go, I got it. All right, so and that includes this KRAS G12D that was not identified initially in the pyrosequencing run. Now, further investigation of some of these discordant calls were eventually, just to summarize, what we ended up realizing was that the Agena was detecting variants that were truly there. We confirmed them using orthogonal methods, and that the Agena was actually more sensitive than our initial pyrosequencing assay. And so the PIK3CA mutation was confirmed. The EGFR insertion, again, it wasn't detected initially using pyrosequencing, but we were able to confirm that not just by running it on the Agena, but we did it using NGS and identified that insertion there. And finally, the KRAS G12D that was not identified by pyrosequencing, we eventually realized that it was probably what we were dealing with was a very, very low allelic frequency variant in the tumor, likely a product of very low tumor cellularity that we were dealing with. Okay, I am at 15 minutes. Some of the other issues, we well, just to look and see how we could modify some of our criteria, we looked at dropping the z-score, dropping the threshold for calling a variant down to 10, and that certainly identified some additional variants at what could be a very low allelic frequency. But ultimately, again, we ended up with the finding that just keeping a very conservative threshold for making a variant call and ensuring that the tumor cellularity was sufficient would be enough to identify reliably our variants of interest. So as far as the summary of our discordances, so we did identify an EGFR exon 19 deletion that was not identified by pyrosequencing. We detected a PIK3CA mutation that was not detected by pyrosequencing. We identified a different exon 20 insertion, which we confirmed. So Agena made the right call in this instance as opposed to pyrosequencing. And then the MassArray called a KRAS G12D, which upon further inspection was of low confidence, and we opted not to call it at the very end, just because it looked like it was a very low variant allelic frequency. So ultimately, we found that if we kept the z-score at a level of 35, we kept ourselves away from any sort of false positive calls. Manual inspection of the 4 discordant samples ensured that 3 of those 4 were definitive calls and 1 of them was indeterminate. And ultimately, if you adjust the z-score to 10, you can identify more variants. but those can be iffy as far as determining their validity, and you might need some orthogonal methods to ensure that you are calling a true variant. As far as costs are concerned, I am willing and able to disclose our internal cost per test or sample. Discussions regarding the cost of the equipment are above my pay grade. Also, these costs are based on certain assumptions and should only be used as an approximation to the costs that may be involved in your lab. So, yeah, this worked out great. No, so thankfully I have a summary slide. So, for comparison, so if we break down the cost, and we can send along these slides. I fully intended for those two tables to show actual data, but some of it got lost in translation. But ultimately, if you look at the totals for them, if we're looking at the NGS assay, TST15, we're talking about $220 per sample. If you break it down per gene, it's about $15 with a turnaround time of 36 hours. For pyrosequencing, you have to do each gene is only $101, but you do it times 3, so that's $303 per sample with a turnaround time of approximately 24 hours. And then the Agena Lung Panel, the cost per sample is actually $112 per gene. It's slightly more expensive than our NGS assay, but ultimately some of the onboarding costs were attractive enough that we went along with it and are currently using it. Now, the question you should ask yourself is, does this fit in your stable? All right, so Agena, following this validation study, the MassArray has become our workhorse. The way our workflow currently goes is when we have a tumor, a lung cancer, a colon cancer, it comes in, and we want to do an initial genomic analysis, we go through the Agena MassArray. And when it identifies a sample, good. When it identifies a variant of interest, then good, we are done. We go no further. If it's negative, then we use some of our additional NGS testing that can look for some of the amplicons that are not included in the initial MassArray assay. And so when you do it that way, you have a fairly cheap, fairly easy to use kind of workhorse that comes in initially. It analyzes your lung and your colon cancer samples. If that's negative, okay, then you look for the more esoteric variants. Then you do your NGS sequencing. Then you go through the whole bioinformatic analysis, and then you identify, and then you see if you're identifying anything a little bit more unusual. So that has been our experience, and it currently is our main workhorse. The catch, so main reason I was reticent to adopt this technology is that while my technicians are running the Agena assay, they are not getting the experience and expertise that I would like them to have in NGS. There is something to be said for that loss of experience. There is something to be said for my techs not sitting down and getting the repetition of running these assays over and over and over again, and being able to analyze an NGS assay quickly. There is some loss of flexibility in the sense that the genes, I find the MassArray to be adequate right now for what is included in the NCCN guidelines right now. But as you can see, there are more genes and more targets being added on a daily basis. And so the flex, as long as they keep coming up with more varied assays, that's not a problem. But to whatever extent they don't, I am stuck. So then I still have to go with an NGS assay, or I still have to send it out. Like I said, gene coverage is sufficient for now. And Agena has been fairly responsive to our needs and our problems with initial issues with the assay. And again, that's for now. So that's that was my initial reticence to work with the Agena MassArray, and I will take any questions. Yes.
For your cost, was that based on a particular batch size or number of samples?
So we use the 96 well plate, and we run about we run about 8 samples per plate. So that is our batch size. Did that answer your question?
Same thing for the NGS?
Yes. So that is a good point of discussion. NGS, that was our estimate once we had optimized our workflow for NGS. But NGS, the cost per gene and the cost per sample can vary a fair bit, depending on, again, your experience with it, and depending on how you design your workflow and how many samples you can load up per chip. So that was our best estimate of the Illumina TST15 assay, a limited gene panel. If you are talking about a more expansive gene panel, like a 500 gene panel, the cost per gene is going to be much lower in those instances.
So, I mean, like NGS would be, say, 8 samples on NGS or 8 samples versus a gene, and that's the cost?
Yes, that is correct.
We'll also have time for questions at the end if you think of some in the interim period. So next I'd like to introduce Dr. Pierre-Jean Lamy. So Pierre-Jean is an oncogenetics specialist in biomarkers of solid tumours following his graduation from the University of Lyon. Yeah, my French is not very good, Pierre-Jean. He joined the Montpellier Cancer Institute in 1997, and in 2007 he became the head of the Department of Biology, where he developed a molecular laboratory dedicated to the detection of DNA alterations in breast, ovarian, colorectal and prostate cancer. At this time, he was also the coordinator of the Montpellier Cancer Institute Biobank and led many translational research projects on prognostic and predictive biomarkers to improve personalised medicine. Since 2011, Pierre-Jean's interests have focused in the area of liquid biopsy as companion diagnostics for molecularly targeted therapies. He's currently the director of the genomics laboratory at Imagenome, which is a laboratory dedicated to development of theranostic tests in the clinical laboratory group Laboratoire Innové.
Thank
I
you.
hope I got all of those French words correct.
Thank you, Darryl. I will try to do my best also in English. Good afternoon, everybody. So the introduction is down, and I can move to my presentation. So I'm coming from Montpellier. This is in the middle of the south of France, so maybe you can know this lovely city. And I'm very happy to be now in Baltimore. This is my disclosure. I have a lot, and I didn't make the addition of the money I get, but I get it. So everybody knows in this room that we are at the era of precision medicine, and that at the era we need to get also the right sample to give the right treatment to the right person. And the right sample is not easy to get, especially in lung cancer. We usually have lung biopsies. This is those teeny things. You can see this picture, and it's very difficult to get it because lung biopsy carry risk for patients, blood clots, bleeding, pneumothorax, pain, infection, fever, pneumonia, etc. And it's also difficult for the patient. Those patients have difficult to breathe, and they need to stop breathing, to stop coughing during the biopsy. And sometimes you cannot prescribe a biopsy for this patient. And for us, molecular biologists or pathologists, it's very difficult to analyze because we get only a low number of cells, and it's sometimes insufficient to make the diagnostic and also to get also the molecular information. And if you look at these results coming from the publication by UR in GTO in 2014, at least 20% of patients had non-evaluable tissue at time of diagnostic. It was the IFUB study. It was a randomized clinical trial. So you know also that in non-small cell lung cancer, it's mandatory to analyze EGFR and other genes. As we have seen, I will go quickly because you know we can prescribe for patients presenting an activating mutation, those TKI inhibitors of first and second generation, but also those patients after treatment can get resistance mutation, especially the T790M mutation, and we have now the possibility to treat those patients with third generation of EGFR inhibitors called Tagrisso. This is specifically designed to be active on the T790M mutation. But now we can also use this treatment and first-line treatment, and the problem is we can also see those C797S mutation. This is a resistance mutation to Tagrisso, and there is also a very recent publication by Oxnard in Thoracic Oncology in the beginning of August, showing that those patients can be treated and can come back to the first line of treatment and treat this patient with, it was gefitinib. So, just to remind you that we have different evolution during the course of the disease. We need to make a molecular reflex testing at the beginning, and then to look at resistance mutation, and also during the evolution of the patient to adapt TKI regimen. And it's very difficult to biopsy those patients several times, you know, so maybe the liquid biopsy could be the good thing. And fortunately, we have those guidelines published last year by the CAPS, ILSLC, and the AMP molecular testing guideline, and they say that ctDNA could be used for clinical setting in which tissue is unlimited and or insufficient for molecular testing. So that's great. Yes, we can. But the problem is how can we do that, and how can we deal with the many challenges we have to face it for CF tumor DNA analysis. And I take this picture, maybe you have child and maybe you use it, you use this book in the US, I don't know, it's several languages, you can use it in English, in France. This is Where is Charlie? In English, this is Where is Wally? The pitch is to find a guy in this picture. This is a very complex figure, and there are many stripes and a lot of people, and this guy has a sweat with stripes. He has glasses also and a hat. So, did you find Wally? So it's very difficult. This is the same with cfDNA. There is a high background, and you have just to find some small DNA fragment specific coming from the tumor. So if you get a very good test, if you have a very sensitive one, you can see this guy, Charlie or Wally, is there. And okay, I picked him, and I'm very happy. So this is the same. So we have. Why do you have this problem? Because we have first pre-analytical issues. We can use different tubes, but we have to avoid dry tubes. Because when you draw a sample in a dry tube, you stress the white blood cells and they release a high amount of genomic DNA. And so this is like a dilution of your specific analyte you want to analyze. So you can use also classic EDTA tubes, but this is the same with a long time. You can keep your samples at least 3 hours before centrifugation and then frozen the plasma. And of course, it's better to use preservative tubes, and we have some data about that to show that this is the best. Because we have a very low amount of analyte, you have to use high volume, maybe to make a double centrifugation to discard also cells that can bring the genomic DNA in your samples. And you have to be very attentive to the kit you have to use for extraction of DNA. Some kits are completely bad, and they miss the cell-free DNA. Those ones are validated. We used the Pierre-Jean kit with a real success. And then the Cobas test, and now I moved with a small company in Montpellier. I'm very proud of that. ID solution test. This is a magnetic beads, and you get a high quantity. Then, of course, after extraction, you have to quantify and qualify your sample. It's very important. You know, the main process for cfDNA is related with apoptosis. This is a release of a nucleosome in the circulation, and that's why we get small fragments. This is the length of the double helix in the nucleosome of 160 base pair. So this is those fragments that are interesting. So you can analyze them with a qPCR, and this is better with, than spectrophotometry because this is very, very small fragment. You can also use a lab shape system or any microfluidic capillary electrophoresis system for quantitation and size analysis. And you will have those electrophoregrams like that. So the good one with this peak here, small fragment. The bad one, you have no cell-free DNA. And the ugly, because you have a lot here of high fragment with a high size fragment of DNA, and this is like a dilution. And I will show you the last technique you can use, a liquid IQ panel developed by Agena for fragmentation, amplifiable copy number quantification, white blood cell contamination, very interesting, and also identity. We have to face also to analytical issues. Of course, we use molecular biology, so if we can use Sanger to analyze cell-free DNA, it's not forbidden, but Sanger has a very low sensitivity, so it's okay if you have a huge quantity of cell-free DNA. It's okay for advanced metastatic cancer with patients with a high tumor burden. But if we want to analyze patient at time of diagnostic during follow-up, to detect early recurrence early, we have to use very sensitive methods, PCR, beaming, digital, NGS, but adapted for liquid biopsy. Otherwise, as my colleague said before, it's around 5% sensitivity, and the MassArray. So I will show, so you have here the reason for why it's very important to have very high sensitive method. Usually with liquid biopsy we get a few quantity of DNA, 10 to 20 nanograms. And so if you want to detect an allele frequency around 0.1%, you just have in your tube 3 or 6 fragments of DNA, specific DNA. So that's very important to have a very sensitive method. So I will show you the UltraSEEK biochemistry we use. So this is the MassArray technology. So it begins like a classic one with a specific PCR looking at the gene of interest you want to analyze. Of course, you have a lower level of mutant template. And the pitch is to make this single base extension, but specific for the mutant allele, using a butynylated terminated nucleotide. So you have this amplification, and you can make a capture of the specific amplification using a MAC bead that is conjugated with streptavidin. After washing, you just have those SBI mutant allele specific, and you can analyze it in a MALDI-TOF system according also the time of flight. So this is the machine, and now this is a panel, so it has already been presented. So looking at 5 different genes of interest in non-small cell lung cancer. And also we use now this liquid IQ pre-analytical control. So to determine the level of white blood cells, to determine the total amount of cell-free DNA, the total amplifiable number of copy of cell-free DNA, the presence of high molecular weight fragment in your sample, and also to authenticate the sample. And there is, in the front of this room, you have this poster, and you can see this poster also at the Agena booth during the AMP this year. So let's have a look at the analytical performance of the UltraSEEK Lung Panel. So this is also a poster that will be presented during the Congress. And for that, 5 different laboratories all over the world were involved to analyze the SERACare ctDNA mutation mix. This is a reference material including 9 cancer-relevant somatic variants, a different allele frequency, 2.1, 0.1, 0.25, and 0.125%, and wild type also. 2 runs were performed per seat, and the limit of detection was determined yielding variant detected rates at least 95% for the targeted variant. So the lowest limit of detection was achieved for EGFR deletion. This is easier to detect those kind of mutation, 0.125%, and the highest of 1% for EGFR insertion. It's very difficult to analyze those insertions, as we have seen before. The overall limit of detection was a median of 0.5%, and the study generates an overall specificity of 99.8% across the more 8,000 negative variants we analyzed. And there were no significant differences in terms of limit of detection of sensitivity across the 5 laboratories. Then we moved to a clinical study comparing the gold standard, like the Cobas test and the MALDI-TOF system, and I presented those results at the MB Global this year in Hong Kong. So the comparison study was a Cobas test V2, real-time PCR. You know this test, looking at 4 exons in EGFR gene, 48 mutations. So the UltraSEEK Lung Panel I presented before, 67 mutations across those 5 genes. And the lung panel study was made with 100 plasma of advanced lung carcinoma patient analyzed for somatic mutation at time of diagnostic, and also at time of recurrence for patient under EGFR inhibitors. Cell-free DNA was extracted with a Pierre-Jean kit, and we made also quality control with a novel liquid IQ panel. It's here a little bit small, but you have also the number of amplicon you can analyze, and you have the identity between the tissue and the plasma. All were concordant and good for analyze. And this is the result for those 100 samples. We found, of course, wild type, EGFR mutant, and you can see here several patients presenting more than one mutation. For patients presenting also T790M mutation, and also a KRAS mutation that is also a mutation of resistance for a patient under TKI treatment. We found also a specific KRAS mutation, and for those patients, 2 were also BRAF V600E positive. If we look at comparison results with Cobas test, first of all, with the Cobas, we get 6%, that means 4 patients with invalid results because of the low quantity of DNA. And we didn't get no invalid results with the lung panel. You can see also we get more EGFR mutant, 49% with the UltraSEEK Lung Panel in comparison with the 40% with Cobas test, and in relation also wild type was lower with the USK Lung Panel. And of course we found it more mutation, 10% related with the other genes that are not analyzed with the Cobas test. If we look more deeply, you have here the blue bar, the total of EGFR mutations. The red bar, it is a T790M mutation. The green bar is for the J719X. Yellow bar, KRAS mutation. Violet for BRAF, and orange bar, the invalid. So you have here the 4th invalid. As you can see, we found it more, of course, mutation. 11 additional mutation with UltraSEEK test, 4 exon 19 deletion, 1 L858R, 3 T790M mutation, 1 exon 19 insertion. We didn't find it with the Cobas test, so it's mutation very difficult to find, and 1 G719A, and 1 L also 861QR. Only 1 exon 19 deletion was detected by Cobas only. So the problem of specificity, this is a real problem. Is those mutations real or not? And you can see here all mutations detected by UltraSEEK only at an allele frequency below the limit of detection of the Cobas test. The limit of detection of the Cobas test was 1.5 to 13% according to the different mutation you look at. So here you have the result I showed you just before. the first analysis we made. For example, if you on board you have the discordance between the lung panel and the Cobas test. If you look at the first one, for example, we found an exon 19 deletion. The allele frequency was around 1% with the USK panel, and the limit of detection for this mutation in the Cobas test is 1.40%. And the 10, for example, here the classical EGFR L858R, 0.28%, and the limit of detection is 4% with the Cobas test. So we are quite confident. It's very difficult to make an orthogonal comparison, but we can say that our results are quite reliable. So finally, I will show you the comparison with tissue, and tissue analysis was possible, available for 80 samples in this study. 33 samples were EGFR mutation negative, and 47 samples were EGFR mutation positive in tissue. In 38 tissue-positive sample corresponding EGFR mutation was detected in cell-free DNA by UltraSEEK. That means a predictive positive value of 80%. And interestingly, 11 tissue-negative samples identified an EGFR mutation in cell-free DNA using UltraSEEK, and we can confirm those results in 10 samples with the Cobas test. So this is now time for my conclusion. UltraSEEK Lung Panel presented a higher sensitivity in a broader panel, of course. And that means different points, a good concordance with tissue analysis. I will show you, and this 80% is quite the same. You can find the literature comparing cell-free DNA and tissue analysis. We found also better concordance with clinical evolution, and monitoring is possible for more patients with the detection of specific resistance mutation, secondary resistance mutation. We found a higher level of mutation at time of diagnostics also that allows TKI treatment for more patients. And we found also mutation for a new mechanism of resistance like pyruvate kinase KRAS mutation, and so it allows us to maybe to change the treatment for those patients. And one point very important is also you can get the allele frequency, and it could be interesting for monitoring, for example, immunotherapy. If you can look, there are some publications regarding the allele frequency of KRAS mutation, for example, that could be a response for immunotherapy. So the need for the future, and I think this is more generally my opinion, we need very high sensitive methods. I think we can agree with that. And multiplex method is very important. We have seen the comparison with NGS, so NGS could be very interesting, but with cell-free DNA we have a lot of negative or invalid results. So this is important to have multiplex method, but NGS is not only is not the only solution for that. And also cost-effective methods. We have seen the different economic analysis just before, but we have to repeat those tests for those patients. So this is a problem to get the money for those tests. It's not reimbursed in France, so it's very difficult for us to monitor those patients. So we need to use a very cost-effective method in terms of money, but also in terms of time. So thank you for your attention. I would like to thank all the patients participating in the study, the clinician for working in the clinic I work with, and also all my colleagues, and especially Dr. Richebourg, Nicolas Lozano, who performed the test very quickly in my lab, and Dr. Sartori from Agena I work with since a long time now. Thank you so much for your attention.
Thank you, both Dr. Castro and Pierre-Jean, or Dr. Lamy. So we heard a little bit about something new in that last presentation, the Liquid IQ panel. And in fact, we're launching that today. So if you haven't heard anything about it before, that's because you shouldn't have. So let me tell you a little bit about Liquid IQ. So this is a panel designed to look at some of the pre-analytical variables and risks in a cell-free DNA sample. Now, in a cell-free DNA sample, there are some difficulties. There's a lot of white blood cells. In 1 ml of blood, in just 1 ml of blood, there's well over 100 million white blood cells. Cell-free DNA is only about 1,500 copies. So we need to be very, very careful to not disturb these guys, because they will very rapidly dilute out what we're trying to look for, which is the cell-free DNA, or specifically the circulating tumor DNA. So this new panel, this Liquid IQ panel, looks at white blood cell contamination. So what can happen, particularly if you're not using cell stabilization tubes, if you're using standard purple top EDTA tubes, is that particularly in the shipping process, if you don't spin those samples down and at the right speed, and transport at the right temperatures within 6 hours, those white blood cells start to lyse. So this panel measures the amount of white blood cell content that is in your sample. Now, another challenge with molecular methods is PCR inhibitors. So PCR inhibitors can be present. This is an example of PCR inhibitors where the same amount of DNA is loaded for 5 different samples, but you can see a reduction in the cycle number, indicating that a PCR inhibitor is present in those samples. So this panel also looks at PCR inhibitors. Within the panel is also a barcode. 21 SNPs as well as 3 sex markers, giving you around about 10 to the minus 9 power of discrimination to allow you to do identity matching. So in these studies where we're now getting a tumour sample and then multiple plasma samples over the course of time, what this panel does is barcodes those samples and cross-references back to the database to make sure that, yes, today's plasma is the same as last month's plasma, is the same as last year's tissue. So it has that barcode in there. On average, 31 specimens is the number of specimens that are mixed up in any given laboratory. Now, I'm sure all of your laboratories are better than that. Maybe. This will tell you whether you are or not, because it's a significant burden on the healthcare system. So what if you can understand the number of amplifiable molecules? Now, we heard about sensitivities down to 0.1%. But let me tell you one thing. You can only detect a 0.1% mutation frequency if you put in at least 1,000 molecules of DNA. You can't detect 1 in 1,000 with less than 1,000 molecules, right? So we have the ability to understand how many amplifiable molecules are present in your sample, whether it's contaminated with white blood cells and the percentage of contamination with white blood cells, whether PCR inhibitors are present, and also that barcoding to prevent sample mix-ups. So doing all of this in a single reaction before you start your liquid biopsy workflow. So this is an example of the report. It's a completely automated data analysis that does all of those various different attributes that I mentioned before. Whether it's passed our QC, so you set a number of molecules or a white blood cell content threshold, so whether it's passed your QC, the SNP markers, the number of amplifiable copies, the percentage of white blood cell contamination. These numbers are a bit low. Up the top there, I think it's 1%, 4%, and then we've got a highly contaminated sample at 90%. So the percentage white blood cell contamination. We also have sizing assays in there that tell you whether long DNA template has been detected. So it flags if there is long DNA template present. Now, of course, long DNA template can come from a variety of sources, right? It can come from necrotic tissue. It can come from infection. It can come from running an ultramarathon. Not that I know too many cancer patients that are running an ultramarathon, but it can come from from extreme exercise. Most importantly, we don't want it to come from white blood cells. So we have this combination of fragmentation analysis as well as white blood cell content analysis to determine whether that troublemaker, the white blood cell DNA, is present. Now, looking at Pierre-Jean's data, because he ran this Liquid IQ panel on those 100 samples. Now, if you remember, in those 100 samples, there were 47 samples that were tissue positive for EGFR, And 80% of those, we had the same EGFR mutation detected in the corresponding plasma sample. Now what I've done here is I've plotted the percentage white blood cell contamination versus the number of amplifiable copies for each of these samples. So 47 samples here. Those in red are the ones that were discordant between the tissue and the plasma. That is, it was positive in tissue, negative in plasma. And those that are a blue dot are concordant between the two samples. Now what we see out of the 9 samples that were positive in tissue but negative in plasma, 8 out of those 9 fail the liquid QC panel because of 2 different parameters. 2 of them, this one here and this one here, highly white blood cell contaminated. This one here is around about 90% white blood cell contaminated. And then 6 out of those 9 have less than 3,000 molecules present. So we set a recommendation of 3,000 molecules such that you can detect that 0.1% mutation frequency. Right? You do have to put in a little bit more than 1,000 molecules. If you only put 1,000 molecules, then you've got a 33% chance that you don't have 1 molecule of the tumour DNA. So based on those 2 thresholds, in fact, if we look at those samples that pass the liquid IQ, and we set a threshold of less than 50% white blood cell content and greater than 3,000 molecules, it would have given us a positive predictive value of 94% and not 80%. And the p-value there is getting close to significant at 0.08 in that discordant results were much more frequent in failing samples than they were in passing samples. So here we're now expanding our oncology and our somatic mutation detection panels to include QC panels to protect yourself from those pre-analytical variables being the cause of a false negative result. All of these panels run on the same instrument, under the same chemistry workflow, and can be run together on the same plate. So thank you for your time today. If you do have any questions for any of our speakers, please speak up.